Hello, AP Biology. This is Mr. Stratton, and I wanted to give you a quick rundown on Week 2's FRQs. So this first question is going to look a little overwhelming, but I wanted to give you some breakdown of this question so that you don't feel quite as overwhelmed looking at this. So again, structure-wise, these are going to be very similar to what you're going to see on the AP test. The first question is 25 minutes, so 10 point there. And the second question is going to be 15 minutes. And when you take the actual AP test, you're going to then be given 5 minutes for each question to upload. So you're going to take question 1, 25 minutes, 5 minutes to upload. Question 2, 15 minutes, 5 minutes to upload. And there's going to be a timer on your screen that's going to turn red at the 5-minute mark to let you know that you have 5 minutes left to upload. So you want to stop writing and upload at that point. So as you're going through these, try to time yourself and be consistent with your timing. Again, this is about preparation, about practice. Okay, question number 1, we've got a diagram here. This diagram is showing cellular response to infection by pathogenic bacteria. So take a look here. Look at all these different molecules we've got going on. It looks a little scary, right? But what I want you to realize is that there's more information coming in the paragraph below. So take a look at this, figure out what you can just by looking at the diagram, and then read the question. So we've got inactive caspase 1, which is then being activated by bacterial infection. That then goes on to activate 2 other reactions. So we've got inactive interleukin being activated, and we've got gasdermin being converted into N-terminal gasdermin. So the active interleukin is going out of the cell, it looks like, and the N-terminal gasdermin is going to the membrane where it looks like it's making some sort of pore. So if you read through the question, that's exactly what's going on. It's going to give you a breakdown of everything that's going on here in that diagram. So skip down to the second paragraph. Researchers created the model in figure 1 using data from cell fractionation studies. Now, if you don't know what cell fractionation is, you probably can sort of guess by the fact that, I'm going to have cell fractions. Okay, so they, in the experiment, various parts of the cell were separated into fractions by mechanical and chemical methods. Specific proteins known to be located in different parts of the cell were used as markers to determine the location of other proteins. The table below shows the presence of known proteins in specific cellular fractions. In other words, they busted up some cells, they separated them out into fractions, and those fractions then are related to different locations within the cells. Like we know, for example, that the nucleus is big and heavy, so that's going to be one fraction. Other organelles are going to be another fraction. The cytoplasm is another fraction. So let's look at the table they give us here. So the whole cell sample, that's the entire cell. Fraction 1, we've got actinase, whatever that is. It's a Krebs cycle protein. So you know where Krebs cycle takes place. You know what's in that fraction. Okay, DNA polymerase was found in fraction 2, as well as NF-kappa B, an immune response protein, whatever that is. Fraction 3, GAPDH, it's a glycolytic protein. So glycolysis, oh, do we remember that? Okay, where does glycolysis happen? Well, you're going to be able to figure out what fraction of the cell that is then. Then fraction 4 has the sodium-potassium pump. Where is the sodium-potassium pump? Remember, it was responsible for maintaining cell water balance and ions and cell polarity. So think about where the cell is working to keep its polarity. All right, so there are 5 parts to this question. So part A, describe the effect of inhibiting step 3 on the formation of pores and on the release of interleukin from the cell. So you're going to want to go back and look at the diagram, find step 3, and talk about the effect of the formation of pores and on the release of interleukin. So you've got 2 points from part A. Part B, it says make a claim about how cleaving inactive caspase 1 results in activation of caspase 1. So you're going to make a claim. That's 1 point. A student claims that pre-infection production of inactive precursors shortens the response time of a cell to bacterial infection. Provide 1 reason to support the student's claim. So in other words, the student's saying that, hey, if they do this first step ahead of time, then it's going to make this faster. So give us a reason why. So 2 points for part B. Part C, a student claims that NF-kappa B protein is located in the cytoplasm until the protein is needed for transcription. Justify the student's claim with evidence. So you're going to have to go back and say, how does a student know that this is in the cytoplasm? Identify 2 fractions where N-terminal gasodermin will be found in cells infected with pathogenic bacteria. So in other words, look up at the fractions there and figure out 2 fractions that would have N-terminal gasodermin. So that part is 2 points as well. For part D, describe the most likely effect of gasodermin pore formation on water balance in the cell in a hypotonic environment. So you have to remember what hypotonic is. So the cell is going to be hypertonic, its surroundings are hypotonic, and talk about what's going to happen when you've got that gasodermin pore that's forming in the cell, that opening in the cell membrane. That's 1 point. And finally, part E is 2 points. You're going to explain how gasodermin pore formation and interleukin release contribute to an organism's defense against a bacterial pathogen. In other words, you're going to get a point for saying, how does the gasodermin pore help the cell defend against bacterial infection? And how does interleukin release contribute to the defense of the cell against this pathogenic bacterium? So go back, read through it, find stuff in the diagram that's going to help you answer that. 2 points there. The total is 10 points, and you're going to time yourself, give yourself 25 minutes. Okay, moving on to question number 2. So for question 2, scientists have recently discovered that cancer cells often contain morphologically distinct mitochondria compared to those found in non-cancerous cells. The mitochondria of cancer cells frequently undergo mitochondrial elongation, which is shown in that diagram there, characterized by a more tubular shape and an increased number of convolutions, which are foldings in their cristae membranes. So you can see that the cell that is cancerous has longer mitochondria with more convolutions, more foldings in the cristae membrane versus the non-cancerous cell. So this is a 4-point question. Part A, describe the reactions taking place in the mitochondrial cristae. So what's going on in the cristae? Part B, explain the purpose of these reactions in a non-cancerous cell. So in other words, what do those reactions from part A do? Part C, predict the effect of mitochondrial elongation in a cancerous cell. In other words, so this cell is cancerous, it's got an elongated mitochondria. What is the cell getting out of that? In other words, how does it benefit that cell, that cancerous cell? And part D, provide reasoning to justify your prediction in part C. In other words, so it's getting something from your answer for part C. Why is it doing that? What is its purpose? Like, what is the underlying scientific principle that the cell is benefiting from this? Okay. So good luck on those questions, and I will be posting the rubric on Monday.